Migrating mesenchymal cells must polarize to form a protrusive front and a retractive rear. Miguel Vicente and colleagues have previously shown that different isoforms of non-muscle myosin II play distinct roles in this process. Myosin II-A forms dynamic actomyosin bundles toward the front of the cell that promote both adhesion maturation and the formation of more stable Myosin II-B containing bundles that accumulate at the cell rear, where they suppress protrusion and establish front-back polarity.
But the underlying question for us was like, what's fundamentally different between these two isoforms? I mean, when you pair them up sequence-wise, they are like 80% homolog. And when you go to the motor domain, they're really similar. When you go to the coil domain, they're really similar. So the very first thing that we did was to line them up. And the only thing that was fundamentally different was the non-helical tail domain at the end of the molecule. So we start looking there.
Vicente and colleagues, led by graduate student Alba Juanes-Garcia, focused on a cluster of serine residues present in the non-helical tail domain of myosin heavy chain 2B, but absent from the equivalent region of myosin II-A. A myosin II-B mutant lacking this motif was unable to form stable actomyosin bundles, and was thus unable to restore front-rear polarity to myosin II-B deficient cells.
And what was really a surprise for us was that the insertion of the same cluster in the equivalent position of myosin II-A would make myosin II-A behave exactly like myosin II-B, to the point of being able to restore myosin II-B deficiency. That was really surprising.
Juanes-Garcia et al. Suspected that this critical serine-rich cluster in myosin II-B might be regulated by phosphorylation.
So what we did was mutate every serine to aspartic, and when we used this phosphomimetic mutant to rescue the shRNA-dependent deficiency on myosin II-B in our cells, what we found is that this mutant was not able to restore the migratory polarity, and actually the cells behave like myosin II-B knockdowns. I mean, they had more filaments, that's true, but those filaments were unable to sustain a stable rear, which suggested that these filaments were kind of unstable, and we confirmed this by FRAP, and this indicated that this was likely a regulatory hotspot.
Indeed, using mass spectrometry, Juanes-Garcia et al. Found that 3 serines in this critical region of myosin II-B are phosphorylated in vivo.
So what Alba did was like make single phosphomimetic and non-phosphorylable mutants, and only one of the phosphomimetic mutants was able to recapitulate the effect of mutating them all, and that was serine 1935. When that serine is phosphorylated, myosin II-B is not very capable of forming like stable filaments.
The phosphomimetic serine 1935 mutant was thus unable to rescue the front-rear polarization of myosin II-B deficient cells, as it was unable to assemble into the stable bundles required to suppress protrusion and define the cell rear. Unlike wild-type myosin II-B, the phosphomimetic serine 1935 mutant localized within cell protrusions, interspersed with myosin II-A, indicating that phosphorylation of this residue regulates both the localization and function of myosin II-B. Protein kinase C-zeta has previously been shown to regulate cell polarization upstream of myosin II-B, and Juanes-Garcia et al. Found that expressing a constitutively active version of this kinase caused mesenchymal cells to depolarize.
That loss of polarity was corrected by a non-phosphorylable 1935 mutant, consistent with 1935 being the most important residue in the regulation of the stability of myosin II-B in the bundles.
Thus, as long as it isn't phosphorylated at position 1935, this serine-rich cluster in the heavy chain of myosin II-B promotes the assembly of stable bundles that define the non-protrusive rear of migrating mesenchymal cells.
So we have 3 things that we are interested in. First, we want to take this into 3D. We want to explore the interplay with other forms of myosin, like we are now looking at myosin I and other isoforms of myosin II and splicing variants of myosin II-B that are only encountered in the central nervous system. And finally, we want to investigate how myosin II-A regulates polarity in other models that are not mesenchymal. We are particularly interested in neuronal polarity and amoeboid leukocyte polarity. Those are the next steps.
For now, though, you can learn more about how a regulatory motif in non-muscle myosin II-B regulates its role in migratory front-back polarity in the paper by Juanes-Garcia et al., published in the April 13, 2015 issue of the Journal of Cell Biology.